I thank the organizers for inviting me to present the work that my collaborators and I completed and published last year. We found that the agent-based computational modeling of glioblastoma predicts that stromal density is central to oncolytic virus efficacy. I have no disclosures to make. So we developed an ex vivo platform to evaluate the infiltration of the oncolytic herpes simplex virus, or OHSV, into glioblastoma. And we developed a computational model of the tumor cell dynamics. And we found that low stromal density is highly predictive of OHSV1 therapeutic success. We validated the findings in a heterogeneous patient samples from brain metastatic adenocarcinoma. In this work, we use the OHSV viral vector called rq-nestin 34.5 version 2. And it's also been used in a clinical trial for the treatment of recurrent glioblastoma. And that was organized by investigators at Dana-Farber and Brigham and Women's Hospital. The vector is neuroattenuated and replication competent, recombinant and genetically engineered herpes simplex virus. That's a mouthful, but I'll explain that. So the vector encodes 2 gene deletions and 1 insertion. The deletion of the UL39 gene for viral ribonucleotide reductase activity is disrupted, and this results in inhibition of nucleotide metabolism and viral DNA synthesis in non-dividing healthy cells, but not in dividing cells. And the deletion of both endogenous copies of the viral gamma 34.5 gene, and that encodes for robust viral growth in an infected cell. And then the virus has an insertion of 1 copy of gamma 34.5 gene, and that's under control of the nestin promoter. And nestin is expressed highly in many features of glioblastoma. And this prevents the viral replication in healthy cells, and it selectively activates this expression in the glioma cells. So the mechanism of action is that the virus replicates in and it causes lysis in rapidly dividing glioma cells. It releases those virus particles after the first event, then it infects the neighboring glioblastoma cells. And this elicits a tumor-specific systemic immune and cytotoxic T lymphocyte response, and thereby killing the non-infected tumor cells. So we leveraged this OHSV treatment strategy with an ex vivo tumor platform, or slice culture, to develop an agent-based computational framework to recapitulate the treatment paradigm and assess key viral and tumor-specific characteristics that determine therapeutic efficacy. We then used the model to generate hypotheses and to investigate the key determinants of the efficacy. So in this ex vivo platform, we cultured fragments. We cultured fragments of the tissue in the presence of PBMCs, or peripheral blood mononuclear cells. And then based on these empirical data, we built a computational model that describes this entire process. And we included such things as glioblastoma cells, stromal cells, infected cells, cytotoxic T lymphocytes, and the T helper cells. And we ran several simulations to develop and improve the model, and then validated it using new empirical data. We found that treatment efficacy is related to tumor cell density, with greater density associated with higher efficacy. So in this ex vivo tumor model, patient biopsy samples or resections were manually cut up and cultured with the PBMCs and maintained ex vivo in these 24 well plates. OHSV was then added to the tissue fragments, and tumor tissue slices were then formalin fixed and paraffin embedded, and serial sections were collected and stained for HSV1, for cleaved caspase 3, for H&E, and these sections were also subjected to multiplex immunohistochemistry to look at T cell and other cell types. So in this agent-based model of glioblastoma patient tumor tissue slices that are infected with the viral vector, the simulation domain modeled here on the left in panel A modeled the entire process of all the agents seen in this legend over here with virus, chemokines, dead cells, CD4 T cells, CD8 T cells, stromal cells, glioblastoma cells, and infected cells. So in this process, the virus would be uptaken by either the glioblastoma cell or by the stromal cell, the pink cell, and the stromal cell acted as a sink where viral replication did not occur. But in the glioblastoma cells, there was proliferation and lysis and the release of more virion particles. And what this did is it activated the chemokine response. So T helper cells and cytotoxic T lymphocytes were then activated by this chemokine gradient and then went on to infect or then went on to kill the glioblastoma cells. So this table of agent phenotypes and rules and cell-cell interactions shows the extent to which we model the processes and what each agent underwent. So for example, the agents are in uninfected cells, infected cells, dead glioblastoma cells, stromal cells, inactive CD4 T cells, active CD4 T cells, and active CD8 T cells. And we modeled migration, lysis, disintegration, viral uptake, proliferation, chemokine secretion, apoptosis. Of each of these cell types, we modeled these different interactions. So this schematic shows how the CD4 positive and CD8 positive T cells were activated and the responses that we end up modeling. So, for example, in this first panel, the CD4 T cells become infected or activated by the infected cells. And this goes on so that the activated CD4 T cell secretes a chemokine that then recruits the neighboring CTLs or the CD8 positive T cells in this chemokine gradient. And then the activated CD4 T cells proliferate. We also modeled how the CD8 T cells attach themselves to the infected cells. And then after a certain number of minutes, the infected cell dies and the CD8 T cell is released from the dead cell. And CD8 T cells increase with proliferation after detachment from the infected cell. So we modeled both the CD4 and the CD8 T cells using a multitude of variables in these models, actually about 45 different variables, ranging from attachment rates and release rates to proliferation rates. This schematic shows how we modeled the sparse and dense glioblastoma cell content. a patient-derived resected glioblastoma was subsectioned into smaller slices, which were then formalin fixed and paraffin embedded and stained for H&E. And then a clinical pathologist scored the fraction of tumor cells, necrosis, immune cells, and stromal cell content. So the two subsections were recreated in our agent-based model and designated as either sparse here at the top or dense here at the bottom, based on these pathology scores. And this schematic just shows a higher magnification detail than the previous slide, showing the difference in the cell content for the sparse at the top or the dense tumor cell sections at the bottom. Now, this schematic of the sparse in A or of the dense in B content was informed by the pathology scores, but a close-up shows what kinds of cells we modeled in each of these configurations. So for example, in panel D, you can see different colors and different sizes of circles. And these indicate the glioblastoma tumor cell, which is in purple, the stromal cell, which is in pink circles, infected tumor cells, as well as the CD4 T cell and the CD8 T cells. So all of these different cell types were modeled in our simulations. So different densities of cellular tumor were then modeled based on the pathology scores of areas denoted in panel A. Here you can see in the H&E stain, high dense tissue and low density tissue. And these were modeled on the left in different configurations, ranging from purely sparse tissue, 0 to 100, or at the bottom, 100 to 0, which is completely dense tissue. We modeled all of this in order to account for the variability and the heterogeneity that we found in the glioblastoma. So this schematic shows simulations of the computational model for fully dense sample over here on the upper left, or the sparse sample on the right, upper right, or the patchy simulation, which is on the lower left. And live cells are denoted in a darker red dotted line, and dead cells in a darker orange, and virion particles in the lighter yellow or the lighter orange. And what we found was that in the dense sections, the live cells drop from about 100% down to 25% content, 25% being over here in the x-axis. And dead cells rise from about 80% content, or to 80% content from around 0. And the number of virus particles drops from an initial lysis burst. You can see this up here around, well, close to 2.5 times 10 to the 7th particles, down to less than 0.5 times 10 to the 7th, a big drop. So, and in this next simulation, the sparse cellular tumor simulation, live cells drop much less, from 100% down to about 80%. And after this initial virus burst, which is lower, you can see, same amount of virus added, but much less than the dense configuration because the virus does not replicate as well in the sparse tissue. And you can see that dead cell content rises from around 0 to only about 20% of the fraction. So very few cells are dying in the sparse content simulation. However, in the patchy configuration, you can see that it's intermediate between the dense and the sparse configurations. This figure shows the validation of the computational model using empirical data. So in panel A, the number of CD4 positive T helper cells and CD8 positive T lymphocytes were measured in tumor slices from 4 different patients. Different colors are represented in the bars, the columns on the graph. So, and you can see that the second patient has a lot more CD4 T cells than the others, and many more CD8 T cells than the other 3 patients. But you can also see that the level of CD4 cells is consistent with the level of CD8 T cells in each patient. So if it's low CD4 in the first patient, it's low CD8 cells in the same patient when measuring CD8 tumor cells or T cells. So panel B shows the nearest neighborhood distances between the CD4 positive or T helper cells and the CD8 cytotoxic T lymphocytes. And this is averaged across all 4 patients. And Ki-67 is indicated by a little asterisk here, but that marks proliferation. And the Ki-67 non-proliferative subsets of cells were noted for all their cell-cell interactions. So this had to be modeled as well in our simulations. To model the glioblastoma cell proliferation, over here in panel C, we used a logistic model that was fit to in vitro cell counts for U87 cells. It's a tumor cell line, because we could not do this well with the cells within the slices, of course. And panel D shows the average viral infiltration in 50 millimeter wide bands in the periphery of the ex vivo tumor slices. And this is measured at 24 hours. And with an initial burst of 5 PFU per cell, using the label of GFP, which was also in our viral vector in order to track the migration of the cells with the viral vector. So panel E shows the chemokines that were estimated from the data. And we measured the density of the interferon gamma or IFN gamma secreted from the cells. And lastly, in panel F, we show an example of high-grade glioma stain for HSV1. You can see that the immunostaining is confined primarily to the periphery and to this area in the upper right here, where the cells are most dense. And the H&E staining you can see, or the blue staining that is commensurate with the H&E staining is nearly negligible in the middle of the slice, because this slice happened to have a very low density of tumor cells in that area. So this shows a range of tumor tissue configurations that we used and generated with the dense and the sparse regions making up the proportion of the total area denoted by the dense sparse fractions here on the lower left. So for different proportions of sparse and dense regions, the fraction of the tumor tissue remaining was calculated after 72 hours. And the color of the bar represents the initial number of glioblastoma cells in this tissue. So for example, 100 to 0 shows a much higher level of glioblastoma cells relative to, say, 1090, which is only 10% of the cells were actually tumor cells. Panel B shows the percentage of glioblastoma tumor cells remaining after 6 days in these simulations for 3 different administration protocols. A single injection into the center of the dense slice, that's denoted in green. Second is a homogeneous administration of the virus into the periphery, that's orange. And finally, the administration of viral injections into each of the center of the dense patches for 50-50 dense patches in the periphery, and what that's in blue. And what we showed is that the total amount of virus administered was conserved across the protocols but that the simulation showed that injection of the virus into the center provided the most efficacy. And that makes sense because these viral vectors are designed to replicate in rapidly dividing glioma cells and not in neurons, for example, and not in stromal cells due to their expression controlled by nestin, the nestin promoter, and the deletion of the viral genes that are necessary for replication within all cells. So, brain metastases. A brain metastatic adenocarcinoma was isolated for ex vivo experiments, and multiple independent replicates were then treated with the viral vector at a PFU, or plaque forming unit, of 5 per cell. It's a very high level of virion particles per cell. So regions were then characterized by a clinical pathologist, and they were marked as either retaining high or low tumor cell density. In panel D, you can see this, and immunostaining for HSV1 and cleaved caspase 3, markers of cell death, and HSV1, of course, is just the presence of the viral vector. In both the low tumor density in the upper row and the high tumor density in the lower row showed the extent of staining and showed the extent of replication in each of these areas. So if you graph this in panels E and F, you can see that in panel E, the infiltration was almost 3-fold higher for the high density tissue relative to the low density tissue. And this is mirrored in the measurements of cleaved caspase 3 in panel F, which showed similar results for the dead cells. So the infiltration, of course, is commensurate with dying glioblastoma cells. The greater the infiltration, the more cells died. So in conclusion, our simulation suggests that OHSV is best suited to debulk large tumors with dense cellularity, not with sparse tumors or sparse glioblastoma cells within the tumor. And that our models predict that this viral vector will be less successful in maintaining remission, of course, in small tumors with sparse cellularity. And that the simulation showed that the ratio of tumor cell to stromal cell content determines the viral vector's therapeutic efficacy. High tumor cell content is best for use with this vector. And that when we validated empirically by testing the model with the brain metastatic adenocarcinoma patient sample in the ex vivo platform, we found that the viral vector infiltration and cell death were highest in the regions of the high tumor cell content as predicted by the computational model, like we found for glioblastoma. So I want to mention my collaborators and support. So, of course, Dr. Cobbs contributed some of the tumor resections, and in the laboratory collected the tumors for the experiments that I also sent off to Mitra Biotech, and they actually processed the tissue in their ex vivo platform. And their team was made up of Unisha Smalley and Aaron Goldman, and then Sean Lawler, who I think is also present at this meeting today. And then these other individuals range from David Goldman and Bill Goins and Nino and Paul Macklin and Morgan Craig were all involved in the computational analysis and modeling of the glioblastoma infection with the viral vector, the replication, and the lysis of it. So we worked together to publish the manuscript last year. And I want to mention the support from the Ben and Catherine Avi Foundation for my role in this project. Are there any questions?